Here's an understatement. Cancer is awful. No real big surprise there. It's also not actually one disease, but rather about 1,000, and each variation can behave wildly different between patients because cancer has a significant genetic component, and varies a lot between people. When phrased like that, it can seem kinda hopeless, but the ways we treat cancer have come a long way over the years, and we now have some new and very different ways to try and treat it. Some of the new techniques I find the most interesting are methods that aim to teach the body to fight off the cancer itself, rather than having to cut you open and melon ball chunks out. These methods are still pretty new, but represent one of the many footholds we found in the fight against cancer. One family of similar therapies modify a patient's own immune system to recognize the cancer cells and kill them. To do this, doctors first remove some blood from a patient, extract the T cells, which are sort of like your immune system soldiers, grow them in a flask, modify them, and then re-inject them into the patient. It's a long process, and there's risks associated with it as you never really know how your system is going to handle the modified cells, so doctors need to monitor you through the whole process. Over the past several months, I've been in Florida visiting my friend Gabriel Licina's lab. Several months ago, he was working on a version of this sort of therapy, and I got to film the first part of the procedure where the T cells are extracted and made to grow in a culture flask. It's a really cool process that uses some very clever techniques, so I wanted to go over it in this video. While I don't currently have access to a mammalian cell culture lab anymore, I do hope to revisit this sort of procedure and explore how other cell types are extracted, like blood-derived stem cells. Also, if you haven't already seen them, I've put out 2 other videos from my time in Florida. The first was my treatment for lactose intolerance, and the second was keeping mammalian cells alive using a special cryogel we made. Links to both of those can be found in the description, and I'll give some updates at the end of this video. But for this video, we'll just be focusing on T cells. There are 2 systems that you'll typically use to perform this extraction. Either you use dynabeads, which are tiny, specially coated magnetic beads that are similar to DNA extraction beads that I showed how to make in an earlier video, or you use the rosette sep system. Right off the bat, of the 2, it's not even a question which one's better. The rosette sep system procedure takes maybe an hour, whereas the dynabead procedure took 4 hours and gave a tiny fraction of the yield. Rather than going through both procedures in full, let's go over the rosette sep system first, and then look at how much more difficult this normally is without it. To start, we're going to need some blood to extract T cells from, and for the sake of the first test, we had a nurse draw some of Gabe's blood into several small vacutainers. The vacutainers contain acid citrate dextrose as an anticoagulant, so the blood doesn't clump up. It has to be citrate-based because otherwise it'll interfere with the rest of the extraction process, so don't use heparin. The blood needs to cool to room temperature, so while it does, we can set up the extraction. We'll be using special Sepmate tubes, which look like falcon tubes but have a small chamber built into the bottom of the tube. We'll need to fill this chamber with polymorph density gradient media, which is just a special really dense liquid. A sterile syringe is used for this, and of course all of the work is done inside a laminar flow hood while wearing gloves. Be sure to spray down the work surface, your arms up to your elbow, and any reagents or supplies before they enter the hood with 70% or higher rubbing alcohol. Fill the tube with 4.5 milliliters of density gradient media by injecting it down the central hole. It'll fill above the chamber, and this is normal. When you spin blood down, it normally separates into several phases. The bottom layer is mostly red blood cells, as they're very large and heavy, so they sink quickly. The top layer is plasma, which is made of water, lots of proteins, and minerals and nutrients. The layer we're interested in sits right at the interface between those layers as a hazy white layer. This layer is all of the light cell types. Things like T cells, which are special forms of white blood cells, platelets, stem cells, and other mononuclear cells. The density media and special chamber helps make this separation clear so it's easy to pick out the layer that you want later. The way the rosette sep system works is by sort of hijacking this process. Once the blood is cooled, it's all added to one large sterile falcon tube, diluted with an equal volume of phosphate buffered saline with 2% fetal bovine serum, and then a cocktail of antibodies that come with the kit is added to the stock of blood. The antibodies stick to all of the cells except the T cells we want, and then a second set of antibodies connects anything that's been targeted by the first set to the red blood cells. This way, when the mixture is spun down, everything other than the T cells are dragged to the bottom with the red blood cells. You can actually get this system to either isolate or deplete blood of almost any cell type, and I hope to use this in the future for stem cell extraction because of how easy this is. In our case, we added 300 microliters of CD4 isolation cocktail to the blood and allowed it to sit and react for 20 minutes at room temperature. Hopefully you've had some bartending practice, because next we need to gently layer the blood mixture onto the density media in the separation tube, ideally without mixing them for best results, so imitate your best layered shot. In this case, we add 6 milliliters of blood solution. Then we put the cap back on the tube and it's ready to be spun down in the centrifuge, remembering to balance the centrifuge with a tube of equal weight on the opposite side. We'll be spinning at 4,000 RPM for 15 minutes. Once it's done, we can collect the top layer by pouring it directly into a fresh, sterile falcon tube in one solid motion. Next, we need to wash off any density gradient media and other contaminants, so this solution is spun down again, but only for 5 minutes this time. This will form a soft pellet of cells at the bottom of the tube. Carefully remove the liquid with a pipette and replace it with 2 milliliters of sterile phosphate buffered saline with 2% FBS. You can do a second wash cycle for increased purity, but for now we'll move on to the next step as the extraction is basically done now. Just to compare, if we were using Dynabeads for this, there would be several more washes, many more incubation steps, a magnetic separation step, and a lot more reagents. And by the end, there was almost no cells left because of how many times we have to spin down the solution. Which, on the night that we did the procedure, quickly turned into a massive headache where all we wanted to do was go home, but always seemed to have 10 more steps left to go. Now, not to totally bash Dynabeads, as we do need them for the next step of the procedure regardless of how the T cells were extracted. They're actually a really convenient way to manipulate growth factors in a solution, because they can be removed quickly using a magnet. They're just a terrible extraction tool for large amounts of cells like this. With the T cells extracted, we need to get them into a flask so they can start growing. So we prepare a fresh culture flask with 10 milliliters of DMEM with 10% FBS, remembering to label and date the flask. Then we can add our suspension of cells. T cells won't just grow on their own and require special activator compounds, which are really just antibodies that tell the cells they need to replicate. Normally this is only really triggered for an immune response, but we can hack that to get them to grow in a dish. All this means is that we add an extracellular communication molecule called interleukin, and Dynabeads with special antibodies bound to their surface. In this case, we need 30 units per milliliter of interleukin and 100 microliters of Dynabead suspension. Be sure to mix the Dynabeads well before use as they settle out during storage. And that's all there is to it. Before the flask is transferred to a CO2 incubator, they're inspected under the microscope and you can clearly see both the T cells and the tiny round Dynabeads. Once they've been verified to look healthy and there's an about equal amount of Dynabeads and cells, the flask is transferred to an incubator and left to grow for a few days. For comparison, here's a flask made using the Dynabead extraction procedure compared to the Rosette set procedure. You can see just how big of a difference in yield this makes. And from here, there's a lot that can be done with these cells. At the time, Gabe was looking at using CRISPR to knock out the PD-1 gene, which makes the T cells much more aggressive. Other groups modify the T cells to produce new antibodies that are specific to the type of cancer the patient has. In either case, the result is that when the T cells are put back into the patient, they should home to the cancer cells and destroy them. Of course, actually getting this to work and play nice with the patient's biology is always a challenge, but I think the fact that such a powerful technique is now possible is a major milestone. So what came of this experiment? Well, nothing actually. The cells grew well, but the next step of the procedure is to genetically modify the T cells, but this particular batch died during the process, so everything had to be started again. But that's just sort of the nature of biology experiments. No matter how long it took you to get to the current step, your experiment could die and you'll just have to restart at potentially any moment. Sometimes it's your fault, but sometimes cells just don't cooperate and they'll never tell you that when you're first learning about this stuff. All it takes is one miscalculation and you've thrown the solution out of whack and all the cells burst. Probably the worst case of this that I ever heard about was my supervisor in university that spent 6 months isolating protein from a rare species of fungus to test it on a special $40,000 antibody goat. But she did the math wrong and it killed the goat, so she had to spend another 6 months extracting more protein. Science is hard and failure is everywhere. The important thing is that you learn from your mistakes and keep going. Before I wrap up, I want to give some quick updates. First, to those wondering about the lactose project, yes, it's still working, and no, I'm not dying and haven't pooped my stomach out. Since the video went out, I've continued to re-explore the world of dairy products, and I can't say I've been disappointed. Though to the just don't drink milk crowd, you're completely missing the point. Milk is in everything, and unless you're explicitly looking, you're eating tons of lactose per day. The point of the mod was to deal with the unexpected lactose that pills won't help with. The fact that I can now actively eat lactose is just kind of a fun side effect. Also, I got my results back from 23andMe and it confirmed that I'm heterozygous for the lactase persistence gene, meaning I only have a single copy that should be functional. Now, in some cases this means you can tolerate lactose, but often it doesn't, so you end up with conflicting genetic and physical results. This is because the lactase gene can be variable based on one of several mutations, only a few of which 23andMe actually tests for. There's also secondary lactose intolerance, which can be caused by an infection and won't show up on a genetic test. So I think this is a piece of data that adds to the picture, but until there are more tests done, it's hard to show definitively that it worked beyond my subjective reporting. Finally, since I could only work on this project while at Gabe's lab, and that lab is currently in flux, and I'm not in Florida anymore, I won't be able to keep working on it for a while. I may do an AMA or live stream talking about the project, but for now this will be the last you'll hear about it for a while. That doesn't mean I won't pick it up again at some point, but for now I'm working on other projects. If you follow this channel for a while, you'll probably notice that I pick up projects and will drop them for really long periods before picking them up again. This is because I'm constantly moving around, so my access to materials, supplies, tools, and locations varies wildly throughout the year. So I bring most of my core supplies with me and then set up or join local labs wherever I land. For those wondering, that's why the various series I've started only have a few parts in each. It can be a while before things cycle back and I can get a chance to work on things again. I'm going to be putting up another video talking about my plans for the next few months and the future of the channel later this week, so be sure to check back for that for more info. And with that, I'll wrap up this video. As always, if you've enjoyed, be sure to subscribe, and most importantly, ring that bell so you'll see when I post new videos, or you won't be served them because of the weird subscription tier thing that happened to the algorithm. And of course, I need to give a big thanks to my patrons. You're the main reason I can keep this channel going, and I really appreciate your support. If you'd like to support the show and help me keep making videos, consider becoming a patron or buying some sweet science-themed merch. Links in the description. That's all for now, and I'll see you next week.